In this video, we will discuss about NK car in clinical trial. Though we have discussed many times about different components of the cars and different generation of the cars. However, the focus of today's video is mainly on this NK car from the clinical trial perspective. So there are many car NK car or car NK that are undergoing this clinical trial. However, uh, many of those NK cars belongs to two main categories. They are either first generation car or second generation cars, most of them. So as we have discussed many times, this first generation car has only one intracellular signaling, uh, that is CD3 zeta, that is most commonly CD3 zeta, and which is used to activate these NK cells. In the second generations, along with this intracellular signaling, that is CD3 zeta, there is another co-stimulatory signals that can be either CD28 or 41BB. And in the third generations, these two co-stimulatory along with this activating signals are present, and two co-stimulatory could be that CD28 and 41BB both can be combined. And so these are the common three common or different types of car construct. However, there is another area on which the current research is going on, that is to make a universal car that can target most of the tumors. In this attempt, the researchers has find out a common trend or common markers in most of the tumors, that is overexpression of this NKGD2 receptors. So the idea is to design a car construct with an antigen that can target this NKGD2 receptors. Along with that, they have intracellular domains, that is CD3 zeta, or it can be used with second generations or third generations also. So this car that is specific to the NKGD2 can be used against most of the tumors and can be act as a universal car. So this is one of the areas on which the researchers are focusing on, and the other areas on which researchers are focusing on is to find out the alternative of the CD3 zeta. So researchers wanted to find out if there is an alternative of CD3 zeta is available that can activate the NK cells better. So in this attempt, the two main common alternatives for the CD3 zeta that has been uh, tested are this DAP10 and DAP12. And what researchers has found out is that DAP10 performs lower than the CD3 zeta, whereas DAP12 performs better than CD3 zeta. So by replacing the CD3 zeta with DAP12 can have better cytotoxicity effect against the tumors compared to the CD3 zeta or DAP10. And in together, what this NK CAR cell does is it will activate the cells, it will enhance cytotoxicity against tumors and cytokine productions. As the NK cells, as we discussed in last two papers, that main mechanisms by the NK cells perform the cytotoxic effect is through generations or through degranulations of the granzyme and perforin, and it will also increase the survival rate and proliferations of this NK CAR. So since they can kill the tumor cells, so less tumor cells are available, so it will increase the survival and proliferation capability of this NK CAR. And the other things is that how the researchers test this NK CAR before it goes to the clinical trial is they test most of these NK CARs in the mice, which is a NOD mice, in which most of the immune cells are not present. And there are two main approaches that they use: the cell line derived xenograft or patient derived xenograft. The cell line derived xenograft is not able to capture the heterogeneity of the tumors, whereas patient derived xenograft is the better approach, and as it can capture the heterogeneity of the tumors and give us the results that are more reliable compared to the cell line derived xenograft. So this is how the NK CARs are being tested before they goes to the clinical trial. So I hope you understood some insight or get to know about some insights about this NK CAR in the clinical trial. If you need to understand more about this topic in detail, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.